director of the Lester and Sue Smith Breast Center at Baylor, who's going to talk a little bit about team science and his own learning as a leader. Matt?
Thanks, everybody. Hi, Rebecca. Sorry you're not with us. Thanks for inviting me. Yeah, thanks for inviting me to do this. Cliff, I really enjoyed many of your comments. Lots to learn about how to make progress in breast cancer through team science. And I have to say, for me, no real formal training in this. It was a question of learning by doing and accepting that you make personal mistakes along the way in many areas, sometimes in medicine and sometimes in strategy and sometimes your relationships with people. But the key thing is to keep going and show humility in the sense that sometimes when things are going well, it's your fault and you have to own that error. So I thought I would go through just a few thoughts here. First of all, this business of translational science or translational medicine. So I thought this frame it just in a couple of sort of questions. One would be a basic science question, and one would be a sort of clinical science question. So this is a basic science question on this slide. On the left is a Circos plot, not from multiple cancers, from one cancer, a luminal B breast cancer that went on to kill the patient. And you can see how complicated that genome is. It's wild. On the outside ring are all those point mutations in coding genes across the genome. And then you have those squiggly lines, which are all the copy number variations, and all the spaghetti in the middle are all the translocations and large-scale deletions that have occurred in that genome to drive the thing on the right, which is a sort of Hanahan, Weinberg sort of classifications of the hallmarks of cancer. So it's a very high-dimensional problem. How does all that genome recoding give rise to all those different aspects of the tumor phenotype? And when you look at something like that, you realize that when we're treating breast cancer, we're literally in the top 2 or 3 millimeters of the ocean, which is a mile deep. We don't really understand what we're treating. Which leads to the next slide, because this is a clinical science question. Which of these many genetic events determine outcome and create therapeutic vulnerabilities? We all know that getting cancer is like a roulette wheel. You know, you can be a lucky patient, you have a resection, and you do fine. And seemingly the same type of cancer with the same appearance down the microscope, you know, you succumb to the disease in 2 years. What is that? And how does that work? And how do we use all this newfound information about genes, about the genomics of cancer to solve that problem? So I just take you through some examples where people have tried to address this, both from the clinical science forward into the, from the basic science forward into the clinical science, but also the reverse direction, which I would argue is perhaps the more productive direction these days. So here's a fantastic example of a bench-to-bedside piece of research, which actually inspired me to go into oncology in the first place. When I was a medical student at Cambridge in the early 1980s, the press was full of these bizarre viruses that cause cancer in chickens, for example. Chicken leukemia viruses. And then a whole host of people, I'm just showing Tadashi Yamamoto here because he was a postdoc at the time, discovered that the chicken virus was actually a homologue of a human gene. So that's an extraordinary finding. And suddenly there was this insight that there were actually genes that caused cancer. And on top of that, of course, the idea that maybe you could drug that gene and make patients better. Hence the entire field, of course, Dennis Slayman has to be there, showing that if you target the human homologue of a chicken virus, you get better, right? And of course, all the clinical investigators on the right would completely obliterate this slide, but I think Dennis does deserve a lot of credit because the amplification event in human cancers, I believe, was discovered by his team, and subsequently the development of trastuzumab. So that's definitely for basic science to clinical science. I don't even know how you define the discovery of BRCA1 and BRCA2. Arguably, that started with clinical science because it started with looking at family histories and noting there was a familial predisposition to breast and ovarian cancer. And Mary Claire King, using very, very careful linkage disequilibrium studies, showed it was somewhere on the long arm of chromosome 17. But it was actually Mark Skolnick in the University of Utah who actually identified BRCA1 because he was managing to study families that had large numbers of family members, which of course helps track the gene through families. And I'm putting Barbara Weber there because Barbara Weber was also intensely working on this problem. And this is actually a quote, bottom left, I thought was rather instructive. Barb's bearing her heart there because she lost the race to BRCA1. And she says, when you're working on these high impact problems, you get more grants, more money, more speaking engagements, better postdocs. It's very glamorous. So of course, I'm disappointed I wasn't the one to identify BRCA1. And then I thought this was very sweet. She said, on the other hand, you can still look around and say, well, you have your kids, you have your health on a scale from 1 to 10, good to bad. This is really no worse than 5, maybe a 6. I thought that was something that I've had experiences like that in research as well. You're working on something and then someone pips you to the post. Of course, so I'd say that's clinical science going into the lab, because not only did Alan co-discover with his team when he was at Cambridge with Mike Stratton, BRCA2, but also he went on to discover the vulnerability that those cells have to PARP inhibition. And I put Andy Tutt there because he's a lead author on that paper now, showing that you can actually give olaparib, a PARP inhibitor, to prevent relapse in patients with BRCA1 or BRCA2 mutated cancer. So back to, from Alan Ashworth's lab back to the clinic. So really, it's all science. It's just looking at things from different perspectives to discover important advances. I put this up here because of the Ellis findings today. It's interesting, the inspiration for Ellis and new SERDs was actually in part because there was a realization the estrogen receptor was mutant, and you might want to target mutant estrogen receptor in metastatic breast cancer. So who discovered the fact that estrogen receptor was mutated? It was actually Suzanne Fuquay, working at Baylor College of Medicine in the 1990s, using plain old-fashioned PCR sequencing. Patient samples were sequenced for the estrogen receptor, and she found this Y537N mutation, which Anke Borg actually worked on, leading to her graduation. That nice picnic photograph is Anke's graduation picnic. For some years after that, because we had only very rudimentary technologies for sequencing, this point that the estrogen receptor was mutant lay dormant as a finding for the best part of, I don't know, I want to say at least a decade. And then along comes next generation sequencing, the ease of sequencing cancers, and then suddenly, it happened to me working on patient-derived xenographs, suddenly you see that estrogen receptor mutants are all over the place. And then a field is born, then companies start making mutant-specific selective estrogen receptor degraders, and then today we find a positive result for these oral drugs where the efficacy looks enhanced in patients who have an estrogen receptor mutation. So a great story. So I titled this slide, Sometimes Important Findings Start with a Whisper, Not a Bang. Right? So that means you have to read the literature and think, and when findings like this come up, they shouldn't have laid dormant for a whole decade. So really, my personal philosophy is this is a continuum, an iterative wheel. You can enter this wheel at the patient level, maybe through the lens of looking at TCGA data. Maybe you have a mouse model that's recapitulating human disease, or maybe you have a drug that you think should be repurposed. You can enter this circle at any moment. But really, the real point is not really you, but it's teams of people with expertise in these different areas. So that's how you put together advances. So let's look at a few fun examples of this. Today, the discussion of intrinsic subtypes permeated, thank you, Lisa, multiple oral presentations. But where did the intrinsic subtype model come from? Well, it actually came from a NCI grant that I was privileged to be part of back in the 2000s called Strategic Partnering to Evaluate Cancer Signatures. And the NCI gave us a grant to basically make a clinical assay out of Chuck's seminal discovery of the intrinsic subtypes in the late 1990s, early 2000s. I put Joel Parker there because he's the very clever informaticist who worked on gene lists combined with measurability lists, because it was all done by PCR, right, to come up with the PAM50 model, which many people were discussing today. And the biggest question now here around the intrinsic subtypes is, should we be treating patients according to ER and HER2 testing, or should we be treating patients according to the intrinsic subtype? Because we're seeing all sorts of interesting diagnostic mix-ups, like basal-like breast cancers appearing in patients labeled as hormone receptor positive, and clearly they're not doing well on endocrine therapy. So how do we translate that into clinical practice is something we need to think about. We're not done with intrinsic subtypes. But of course, you know, this whole, the roles of academia in keeping, let's say, the commercial world honest is an incredibly important one. On this slide is work with Ivana Sestak and Mitch Dowsett in London, where they've compared all these prognostic signatures, you know, on the same samples, and to drive evidence around which, are there any difference between these tests? Any actionable differences between these tests, and I think that that kind of academic activity also needs to encourage. Again, enormous team science here, because of course it involves samples from an adjuvant endocrine therapy study, TransAttack. And then, you know, and that's all fine and good, but of course, you know, gene expression profiling is a very superficial look at the cancer, amazingly superficial when you start thinking about those circus plots that I was showing it earlier. When I went, I mean, almost the sole reason I went to WashU in St. Louis is because I met Elaine Mardis, and she told me she was doing whole genome sequencing, and I said, "You're doing what?" "Oh yeah," she said. "The human genome is about 3 billion base pairs long, and we can get about 20 to 30-fold coverage." So I said, "You're taking a cancer and you're getting 100 billion base pairs worth of information from it?" And she said, "Sure." I said, "I'm moving. Sorry, Rosanna, we're taking the kids to St. Louis." And then when you're there, you meet through Elaine her team. And at the time, the person who I interacted most is on the left there, Lee Ding, who is probably one of the most efficient scientists I've ever met. Lee, if you're listening to this, I really mean that sincerely. She has amazing talent of taking complicated stories with all this sort of clinical information and distilling it down into the package of information you see there in that article. And when you talk about leadership, Lee really, along with Elaine, are remarkable leaders. But then, of course, that question of how on earth do you make sense of all that data? Well, you can make sense of that data by looking at outcomes. I mean, the problem with the TCGA effort, there really wasn't very good outcome, there wasn't controlled treatment. So here, Obi Griffiths and Meenakshi Anurag looked at that question by collaborating, and Torsten's face should really be on there, a series of samples from British Columbia where they're all hormone receptor positive cancers being treated in British Columbia. He called it the TAM series. There were little bits of tissue left over from hormone receptor testing when it was done by radioimmunoassay, if you can imagine such a thing. And so Torsten kept all these in drawers and drawers and drawers at University of British Columbia, and we came out, we did sequencing of all the recurrently mutated genes that were known of at the time to find out which ones were prognostic. And that exercise gave us a priority list of things you should work in the lab, which gave rise to this finding. So this is a different team now. So I talked to Eric, who's a RAS expert. I said, "Eric, we've got this is a very interesting finding, is that if breast cancer has a mutant NF1 gene," so I'm not saying the patient has neurofibromatosis, it's just the cancer has deleted the NF1 gene. If they have deletions in the NF1 gene, in other words, they have frame shift and nonsense mutations, the patients do terribly in the TAM series. What's that about? Oh, he said, well, we can look into that. We can take some hormone receptor positive cells and knock out NF1. And if you do that, tamoxifen becomes an agonist. So actually, you're giving the wrong drug if they're NF1 depleted, because you're actually stimulating the cancer, not suppressing it, which explains why they did so poorly in the TAM series. But secondly, it gave us models to find out how you would treat NF1 depleted breast cancer, and that's what given rise to this combo match trial with Boralim. The unsurprising answer, we can get complete regressions in patient-derived xenografts of ER-positive NF1 depleted breast cancer with a combination of a SERD, so you don't have the agonist effects, and binimetinib, a MEK inhibitor. So it's a mechanism-based therapy, and this trial is going to be opening very shortly. So that's another different kind of translational medicine, going back to the bench to study something that's been shown to be important in the clinic. For patient outcomes. Another thing that I think shows team science is this whole field of patient-derived xenografts. So when I was an assistant professor, we were all using MCF7 cells if you want to work on estrogen receptor positive breast cancer. I once did a literature search just trying to find out how many papers there have been on MCF7 cells. It's something like 50,000, an extraordinary number. But remember, MCF7 cells, that's only one patient, right? So I said, well, why? So there were some papers on the fact you could directly xenograft breast cancers into immunocompromised mice. Charlotte Cooperwas, I think, is one of the people you really need to credit there. So when I went to WashU, I met Shun Li, and I said, who's a very talented mouse surgeon. And I said, well, let's just graft all these patients I'm seeing into the mice. And that gave rise to lots and lots of models, now called the WIM series, WashU Human in Mouse. And I was talking about this to Richard Wilson, who was working with Elaine. I said, you know, you got a bit of pilot money from the NCI, from your NHGRI grant. Can we sequence some of the whole genome sequencing, some of these patient-derived xenografts? And the bottom line was a very interesting finding. So we had a xenograph from the patient's breast, a xenograph from the patient's brain, same patient, right? And then we had the human samples that were the progenitor, and we did whole genome sequencing of everything. And the interesting thing was, even though the PDX, even though it was derived from the breast, looked exactly like the brain metastasis. So in other words, what we were studying in the mouse was the metastatic clone, giving rise to this term, patient-derived xenografts are really metastases to the mouse. Isn't that a cool result? So none of that would be possible without those clever genomics people, without the wonderful Shun Li, who did all the mouse engrafting. And then, of course, from the tiny beginnings of PDX research in the early 2000s, 20 years later, we've got these massive efforts, the NCI PDX Net project, patient-derived xenografts in every disease you can think of. We're doing genomics in all these models. We're doing proteomics in all of these models, and even relatively large-scale clinical trials in these models. You know, 50, 60 models treated with the same drugs. That's Mike Lewis's contribution there. And Alana Whelm, of course, gets quite a lot of the credit for popularizing PDX with her Nature Medicine paper in 2009. And the last thing to talk about is you've got to keep innovating and keep innovating and keep innovating and stay up with the technology. Here, I'm crediting the Clinical Proteomics Tumor Analysis Consortium at the NCI, just showing a couple of key players, Kelly Ruggles from NYU and Steve Carr from the Broad. Now we're layering the RNA and the DNA information to genome-wide proteomic information and genome-wide phosphoproteomic information and acetylome information. Another team, different sets of technical experts, but really trying to link it to patient outcomes is the key here. So this slide, it shows that we're really in a new phase of deep dissection of cancer using core needle biopsies from patients, either metastatic or early stage, so we can get to the kinds of answers we really need. Patients die because you don't know what's wrong with them. And so that's the objective, I think, for the next phase. So none of this would be possible without incorporating the teams of patients who are involved. Sometimes they just participate because they provide a sample, sometimes they're on a clinical trial, and sometimes because there are advocates. These are all wonderful patient advocates from the breast cancer spore at Baylor. And so rules for success in translational research, Build teams to address difficult, high-impact, unsolved problems through the application of new technology. Don't keep iterating around the same old technology. Grab the new technologies and make them work. And then, as a team leader, don't define success in personal terms. You'll notice my photograph didn't appear in any of those examples I gave, but stress the success of the entire team. And then the key is finding creative partnerships between clinicians and lab scientists. I mean, I think about you and Chuck Peru, for example, Lisa. What a wonderful example of a productive relationship between a clinician scientist and a basic scientist. And focus on improving patient outcomes. Open and transparent partnerships with commercial entities are essential, but many of our research is not commercial in nature, and that has to be encouraged. And we could give endless examples of how clinical science has made progress with translational medicine, and it's been nothing to do with something that was commercially valuable. And then find these mechanisms at the NCI and other entities that encourage institutional collaborations. I noticed Stand Up to Cancer, for example, have dedicated programs to encourage team science. Look at those RFAs, take advantage of them. If you build a good team, it will get funded. And I'll stop there. I may have gone too long, but I hope it was helpful. Thank you.
And I think while you're standing here, and Cliff, before we move to the panel, there's a couple of questions. The first is a comment, which I think is a very valuable one, which is throw in patient advocates into this bench to bedside and back, and you make even faster progress. Which I don't think anybody would disagree with that.
Oh, I think I did have a slide on that on my defense.
No, but I think it's absolutely true. I mean, the patient partnership, particularly as you're talking about the integration of human-derived samples, I mean, this is crucial, right? I mean, a lot of the acceleration is because of patients being willing to donate samples to undergo research, biopsies, and the like. So I think that's...
If I could just pick up on that while you're reading.
Please do.
It.
Yeah, go ahead.
So the mantra for us is there's no more important research for breast cancer research than patients, their outcomes, and their biospecimens. And the other one, I always tease the graduate students, what's the best model for humans with cancer? Humans with cancer. We need to study patients first and then take those findings into the lab. There's fewer and fewer examples where the reverse works.
Definitely true. And I think, I'll just use as an example for whoever wrote this, I think, at my own institution, Patty Spears, who's really an amazing leader in this realm, one of the things that her group does is challenges our basic scientists to be able to give an elevator talk on what they do to a patient advocate in a way that doesn't make the patient advocate say, huh, that doesn't make any sense to me. Explain to me why this is important. Carlos.
The question, is this on? Yeah, the question about the advocates suggests to me that something to think about is that, again, building on what Matt said, the bidirectionality from bedside to bench and back. I would argue that today we have to also think of the community. It should be probably something like bedside, bench to bedside to population science to community and back. And so every now and then we can identify problems in the community that not studied in a clinical trial that are worth studying in the laboratory. Just my thought about it.
No, I think it's...
Yeah.
Well, I think what, you probably say in sort of scientific terms, you have to make impact at the population level, right? Yeah. It's, and that's I think us as sort of translational medicine folks, we want to come up with things that will reduce the death rate from breast cancer and the incidence rate from breast cancer in the entire community, which means in the end, that's your metric. And so you have to go out into the community to engage them so that, advances are adopted and outcomes are improved at the population level, not just, say, a few patients in a clinical trial.
Well, and it's worth noting that at the major medical centers and academic centers are frequently the patients that are seen in those places are oftentimes not that diverse. And so to your point, as you get into the community, many of the challenges that we face actually are not well represented at the mothership.
Yeah. No, I think there's an opportunity to study in basic science laboratories of some aspects of that encompass disparities of care. You know, bringing those aspects of those disparities at the community and study them at a molecular genetic level in laboratories implies a bit of a change in culture in medical centers, but it's something we need to do more.
It's very dependent on how your hospital's set up. I'd be interested to know how it works in Singapore. So at Wash U, Barnes Jewish Hospital was the catchment area hospital for the underserved. So, you know, the lady that works on the corner shop who's having trouble pay for her insurance was sitting next to a wealthy lady from Ladue in the same clinic. And that speaks to the socialist in me. I thought that was absolutely fantastic. Yeah. In Houston, we have a safety net hospital. It's called the Harris Health System. And the patients with limited means are treated in another place, which I don't think is ideal, except Baylor faculty go there and staff it. And also MD Anderson staff, they have their clinics within Harris Health. And that does give us access, the academics access to the patients you're referring to. And many of our xenographs and many of our clinical trial participants do come from that population. So we're rather successful at that at Baylor, just because you have to have your team in those kinds of hospital. I know that does also happen, for example, in Chicago. I'm not sure how it works at UNC, but you know, I think as a program, you have to think hard about how you study that population and make sure you can do that and have advocates from that population.
Exactly.
Yeah, the only thing I'd add to that is my health service research colleagues, I'll use Katie Reeder-Hayes, who I don't think she is here right now, but as an example, say for all those advances that you all guys all come up with, your fancy new drugs and your biomarkers, until you show that that's actually getting out to the community and that dissemination is happening effectively and patients are actually getting it and able to take the drugs, you know, it really matters.
Yeah.
Which is a, you know, I mean, that's the reality check people and our colleagues is a huge part of that getting to the community element. You know, there's a large fraction of patients with HER2 positive disease don't get trastuzumab, don't get basic anti-HER2 therapy, like a surprisingly high number. Mass communications era, but yet in medicine, we are behind in terms of global communication and global collaborations. Rebecca, this is speaking to you. And if nothing else, COVID-19 advanced this to a new platform, and isn't it time to take it to a new level, especially in academia? Would be interested in your thoughts. Cliff, do you want to take the first crack at this one?
I'd love to, but I'm not certain I even understand the question. So the world is connected. Social media is a platform. Your last comment about actual implementation, I think, relates to this a little bit. If the challenge is how do we raise the level of collaboration and advance the cause, I think that's exactly right. I will, however, I will caution in one way, not to disagree, but to be a little bit provocative. What you don't want in a research endeavor, in my opinion, is everybody drinking the same Kool-Aid. You don't want a world with one set of goals. You want 100 strategic plans. You want 100 diverse goals until you're narrowing in on the cure. So contrast that with COVID. The initial effort was obviously vaccination, and the only question was the tactic, which vaccine pathway to take. But we're not there with cancer in general. We're not there with breast cancer in general. So I would worry a little bit about having a unified research theme, if you will, or goal for our community. And I would worry that one platform would be dominated and might drive people in that direction. So that's my only social caution about that. Sharing the results, on the other hand, and keeping everybody up to speed, I think that's an important shared responsibility. I don't know if that answers the question or not.
Cliff, doesn't that, I mean, what you say is very true. Doesn't that then emphasize the importance of inter-institutional collaborations? Because in another institution, you're going to meet people who have a completely orthogonal view to you, and you're going to discover that, in fact, you didn't understand something you thought you understood well. And I don't know how many times I've had that experience. So, yes, I mean, that's probably